Thank you very much to the organisers for inviting me. I'm honoured to be able to give one of the lectures at the conference. This is my working title. The title in the programme is basically the same, which is the modelling and measuring proton movement. And my interest and my group's interest for many years in science has been the science of proton regulation inside cells, particularly cells of the heart. And I'm sure that it should be self-evident why there is this interest. And that is that tissue function, and today's meeting is about tissue function, integrated systems, is powered by metabolism, which supplies, of course, ATP, which supplies the basic functions of the cell. And the problem with that is that metabolism generates end products, and one of the principal end products of metabolism is acid in the form of hydrogen or protons. And acid feeds back and modulates tissue function. That's because protons are chemically highly reactive, particularly with cellular proteins, changing their folding and hence changing their function, which means that hydrogen ions are important modulators of biological function. And classically, if we're dealing with the heart, one of the most famous examples of this modulation is work published by Gaskell 130 years ago, in 1880, in the Journal of Physiology, who showed that acidifying an isolated frog heart very rapidly reduced its contractile function in a reversible manner. And interestingly, this kind of acidosis is precisely what happens during myocardial ischemia, and the acidosis is one of the principal causes of contractile failure under those conditions. This then means that if you want to maintain cardiac function in a steady state, you have to maintain the proton level. So what we are dealing with is a system of waste disposal, and hence this is an extremely fundamental mechanism which is not unique to the heart. It's true, for example, for all eukaryotic cells. And what I'm going to show you today is a combination of mathematical modeling and experiment that's allowed us over the years to unravel this proton waste disposal system in the heart. And I'll show you that we've used mathematics frequently as a tool to define and derive important parameters of the system. And we've also then checked that we understand the interaction of the various components of the proton control system by trying to produce integrated models. Some definitions. Of course, when we say acidic, we mean high hydrogen ion concentration. And the hydrogen ion concentration is measured in a logarithmic form, on a logarithmic scale, the pH scale. And cells are around about pH 7.2, which corresponds to an intracellular concentration of about 17 nanomolar, which interestingly is about the same level as the concentration of calcium. And to give you an idea of the sensitivity, why you've got to control the proton level, you get a 30 to 40% inhibition of contraction if there is a rise in the proton concentration by just 30 nanomolar, if this is uncompensated. And we now know the reason for this. It's principally because hydrogen ions prevent the binding of calcium to troponin C and hence prevent myofilament interaction. And of course, we now also know that acidosis affects calcium signaling and can trigger electrical arrhythmia. So this is a fundamental system which lays the foundation for the efficient function of all the other systems that are operating within the cardiac cell. And if you want to know how it works, well, you could work on the whole heart, but of course there are about 500 million myocytes in a human heart. The way we do it is we go through a multi-scale system. We go down to the level of an individual cardiac cell. This is a ventricular myocyte, which can be isolated enzymically. And if we're interested in proton control, we measure intracellular pH in that myocyte, and we can do that using fluorescent probes. We use the pH-sensitive dye carboxy-SNARF1. So if you load that into a cell and you measure the intracellular pH, in this case it's about 7.0, you can perform a number of experimental tricks that load protons into the intracellular compartment, the cytoplasm. And when you do that, you find over the next few minutes that the proton concentration relaxes back down to control levels. So we have acid up and alkaline down. And this relaxation is caused by the extrusion of those protons from the cell by proteins that are expressed in the surface membrane. And the two relevant proteins that are active under these conditions are a sodium hydrogen exchanger and a sodium bicarbonate cotransporter. This extrudes protons, this brings bicarbonate in, which neutralizes protons. So this is a pH control system. Now these transport proteins are activated, they're switched on by hydrogen ion concentration, which means that as one reduces the pH experimentally inside a cardiac cell, then the population of transporters, and there are many thousands of them in the surface membrane, becomes increasingly switched on, so you get an increasing activation of acid efflux from the cell. And this then produces the relaxation. There's also a system which is switched on by excess base, in other words, by alkalinity, and these then import acid into the cell, and again they're switched on by a change of pH. And the switch, the molecular switch, we're beginning to look at that in our own work. The molecular switch occurs when protons titrate sites on the cytoplasmic part of the protein in the surface membrane, as well as sites in the transmembrane domain. And the thinking is that there's an interaction between these two moieties that then switches on the system and extrudes acid or base from the cell. Now, we've worked on this for a number of years, and we determined the pH-sensitive kinetic characteristics of all the transporters that contribute to pH regulation in cells, and putting them together in a computational model produces the transients that we've just been looking at in the previous experiment. And the experiment matches the model reasonably well, which suggests that we may well have identified most of the elements that control the pH, at least of a given single cardiac myocyte. The problem is that kind of model is zero-dimensional. In other words, it doesn't take account of the geometric space of the cell, and it certainly doesn't take account of any spatial aspects of intracellular hydrogen ion diffusion. And this is where the plot thickens, because in fact these transporters control the pH in the cell only because they're coupled to the cell through a diffusive process. In other words, when a metabolism generates protons, they have to diffuse to the surface of the cell to be extruded on a transporter. So we spent some time looking at proton diffusion in cardiac cells, and it was quite a surprise to us when we started that no one had made direct intracellular measurements of proton diffusion. And when we did, we got a number of surprises. The way we perform the experiment, at least here, is to load a pH-sensitive dye into the cell and image the cell confocally, and then uncage hydrogen ions from a caged compound. And we use nitrobenzaldehyde incorporated into the cell. And we uncage the protons repetitively, once every 10 seconds. And as you do that, you see that the proton concentration builds up in the proximal region of the cell. And we're uncaging using an ultraviolet laser and uncaging spatially just at this end of the cell. And then we watch the protons diffuse down the length of the cell. And we find it takes about 30 seconds before the proton level builds up in the downstream region. In other words, it takes 30 seconds for the protons to diffuse a distance of about 60 or 70 microns. Now, this is very low proton mobility. When you apply models of diffusion to determine the diffusion coefficient, you find that the coefficient is somewhere between 100 and 700 times lower than the mobility of protons in water. So we're not dealing with a free diffusion. Furthermore, the diffusion coefficients that we calculate from these diffusive models are not constants, as one would expect for a simple diffusive reaction. they decline as intracellular pH becomes more acidic. Now, this very low proton diffusion means that the pH within cardiac cells is not constant. Under a variety of circumstances, it's frequently been assumed that the pH is uniformly 7.2. Well, that's true at rest, but if you stimulate acid extrusion, for example, on sodium-hydrogen exchange, I wonder if we can have the lights down so I can see. I don't know where the lights... thanks very much. If you stimulate acid extrusion on sodium-hydrogen exchange and then image the pH at the end of a cardiac cell, you can see that the pH just under the membrane is more alkaline than the pH deeper within the cardiac myocyte. And there can be a significant pH gradient. And the reason for this is, as the protons are being extruded, they diffuse from the bulk to the submembranous space. And because proton diffusion is slow, this sets up significant gradients. Similarly, if you expose regionally a cardiac myocyte to an acidic extracellular molecule, in this case 20% CO2, the CO2 enters the cell and acidifies this region, but we set up a very large pH gradient which is stable for many minutes down the length of the cardiac cell. And this occurs because of the low proton mobility. Now, what causes it? It clearly isn't simple diffusion. In fact, it comes as a surprise to many to learn that protons can't diffuse in eukaryotic cells in significant numbers. That's because they're so highly buffered as a protective mechanism. To give an example, if you had 400,000 protons inserted into a cardiac cell, you would buffer those protons and you would be left with an increment of just one free hydrogen ion. And that means that free diffusion of protons is impossible. And the problem is that most of those 400,000 protons are buffered on fixed buffer sites, which are histidine residues on proteins, which are effectively fixed within the cell. Under those circumstances, protons wouldn't be able to move in cells at all, and the whole system of pH regulation would grind to a halt. But it doesn't grind to a halt because protons are shuttled spatially within cardiac cells on mobile buffers. Now, these are low molecular weight buffers containing histidine that can pick up hydrogen ions in the cytoplasm. The buffer then diffuses spatially. The proton then is released and is pumped out of the cell on a transporter. What are these mobile buffers? Well, the ones in cardiac cells, and I think now we are coming to the conclusion in most other cells, are histidyl dipeptides. There is a collection of mysterious compounds called carnosine, anserine, and homocarnosine that seem to fulfill this function, and they have relatively low molecular weight of about 200. There's also a carbon dioxide bicarbonate system. The bicarbonate can pick up a proton, turn into CO2, which diffuses spatially, turns back into protons, and is then pumped out of the cell. These are the internal transport system for hydrogen ions that are generated metabolically. Now, how do we know that the buffers are conducting protons in this way? Well, this slow rate of diffusion is set largely by the molecular weight of a solute. And if you analyze using mathematical diffusion models this sort of data, you come to the conclusion that the hydrogen ion is diffusing as if it had a molecular weight of about 200, which of course is the molecular weight of the histidyl dipeptides. Furthermore, if we best fit the decline in the diffusion coefficient of protons as pH is acidified, we can best fit it using a buffer population divided into two moieties, a fixed buffer population and a mobile buffer population with different mean pKs and different concentrations. And under these conditions, what you find is that when you acidify the intracellular compartment, the capacity of the mobile buffers to carry protons declines, and the capacity of the fixed buffers to prevent the diffusion of protons rises, and this then produces the decline in the apparent diffusion coefficient of protons. So protons aren't diffusing, they're latching onto buffers, and the buffers are diffusing, and it's the pK and the concentration and the size of the buffer that decides the movement of the hydrogen ions in the cell. And when we code this sort of model into an experimental situation where we stimulate acid extrusion across the surface membrane, then the model predicts this intracellular pH gradient that I showed you previously and is on the left-hand part of the slide here, and it predicts gradients of about 0.1 to 0.2 of a pH unit, which is about 30 nanomolar. And remember, that will halve contraction, that will decrease contraction by up to 40%. So it means that cardiac cells are frequently subject during acid loads to very large intracellular pH gradients, pH microdomains. Now, the story doesn't stop here. There are 500 million myocytes in a human heart. They each have the apparatus for pH regulation. But are they all independently regulating intracellular pH and disposing of their waste in an independent fashion? Or is there a system that coordinates the waste disposal system? And the answer is yes. And this consists, at least in relatively small tissue domains, of gap junctions. If you take a pair of myocytes which are joined by a gap junction and you uncage protons photolytically in one of the two cells, you can then see after a time that protons move into the neighboring cell. So here we're uncaging protons photolytically. The concentration builds up in this cell, and after a period of time, the concentration builds up in the downstream cell. If you put on gap junctional channel inhibitors, you block this transmission. If you look at the magnitude of the transmission, it's enormous. I mean, we can get fluxes which are much larger than the fluxes across the surface membrane of the cardiac myocyte. If you analyze the permeation properties of this transmission using diffusion permeation models, you come to the conclusion that the proton is permeating as if it were a solute of molecular weight about 200. Permeation through gap junctions, again, is determined largely by molecular size, which suggests that the protons are not diffusing through the gap junction. It's the mobile buffer that's carrying the proton that's diffusing through the junction. And that's feasible because gap junctional channels are hexameric assemblies of connexin proteins, largely CX43, which are assembled around a central core, which is a pore of diameter about 12 angstroms and can take quite large molecules up to about 800 or 900 molecular weight. So the mobile buffers, which are the dipeptides that I described to you, quite readily will pass through this channel. And because the mobile buffers collectively in cells are present at a concentration of about 20 millimolar, then you can get very large fluxes of acid between cells. And this is useful because it tends to even out any small metabolic differences of pH between adjacent cells. So this is a mechanism of spatial pH regulation which is producing an intracellular pH syncytium within cardiac tissue. Now, the interesting thing about it, and I can't go into details, is that the connexin channels are gated by protons. If the pH level within a cardiac cell at the level of the junction is reduced to a pH of 6, the junctions close. Now, this has been known for about 30 years. We also found quite recently what was not known is that there is a symmetry in this system. If you raise the pH in cardiac cells at the level of the junction to excessively alkaline levels, the channels again close. There's a biphasic system. And this seems to be a gating system. We've done some molecular biology on these channels now, and this seems to be a direct interaction of protons with the connexin channels, where protons can either activate and inactivate the channel. And the presence of these two processes mathematically predicts a biphasic permeability versus pH relationship. Now, why do we think this is important? Well, what it means is that when the pH is in the normal physiological range of, say, 6.9 to about 7.2, then the permeability of these junctions and their control by protons is optimized, and there's a large fraction of the channels is open, which means when there's a local acid load, it can be dissipated into neighboring myocytes through the open junctions carried on the mobile buffer. So this is a spatial dissipative control. If we now have an acid overload, such as you might get during myocardial ischemia, however, the pH would then drop to about 6. This closes the junctions, and that would tend to seal the acid in the affected cell, which may be a protective measure to prevent the acid from spreading. But under those conditions, the low pH activates the transporters and exports the excess acid out of the cell. And we have used a three-dimensional diffusion reaction model based upon mobile buffers to analyze the relative importance of this junctional traffic versus sarcolemmal sodium hydrogen exchange surface membrane traffic. If we have a central acidified region in what would be a tissue now, if we drop the pH in that central region, so we're moving left along the pH axis, what happens is that we can track the departure of protons from that central region. And we find that the protons depart mainly through the junctions in the physiological range. That then closes as the pH goes very acid, and the protons then mainly depart across the surface membrane. So it's a beautiful coordinated system. And in order to assess the importance of the system, we have to apply computational models. And we've now tested whether or not this is true in a true multicellular cardiac preparation. We've used cultured strands of neonatal cardiac myocytes. We joined forces with Peter Cole's group, who were producing these sorts of pattern growths of neonatal myocytes. We measure the pH in the strand, and you can see each individual myocyte is about 60 microns in length. And they're joined end to end and side by side by gap junctions, which can be stained fluorescently and are shown here. These are the strands viewed at low magnification. If we load the whole multicellular strand with SNARF, we can image it for pH, and again we can uncage protons in the middle region of the strand, and we can watch protons move down the length of this multicellular preparation. And you can see as the protons move from the site of injection, we get a bell-shaped diffusion mobility relationship. So it's diffusing down cells across junctions, down the next cell and across the next junction. If you block gap junctions, and we do this pharmacologically with glyceretinic acid, you then find that basically the protons that you inject here remain within the central region, and we get virtually no movement. And that's shown here. You get a much greater acid load in the central region when the junctions are inhibited. And this is a way of showing you directly the spatial regulation caused by gap junctions. If the junctions were not there, and you get a metabolic local acid load, you get a very large accumulation of protons, which would shut down cardiac function. But in fact, what happens is that the protons flux out into neighboring cells through the junctions normally, producing a much lower level of focal acid loading. Interestingly, when we analyzed the mobility of protons in these neonatal strands, there was a surprise. In the absence of bicarbonate and Ca2, which is part of your mobile buffer system, so we now only have the dipeptide intrinsic mobile buffers within cardiac cells. In the presence of just those, we find that the mobility in the neonatal strands is about 10 times lower than the mobility of protons in adult cardiac cell pairs. And these are ventricular cell pairs. In other words, protons hardly move in the absence of bicarbonate in neonatal strands. If you add bicarbonate, mobility is restored, as it is increased also in the adult system. And this suggests that neonatal cells may lack some of the dipeptide intrinsic mobile buffer system, which means that its ability to shuttle acid down the length of the strand will be severely impaired. And we've now developed an assay for the mobile buffers in our cardiac cells. It's an absorbance assay based on fluorodinitrobenzene, and it measures low molecular weight histidyl dipeptides. And we find that the dipeptides are much higher in adult ventricular tissue than they are in neonatal ventricular tissue. So this is a nice validation of the model where we find that mobility is defined by the presence of the intracellular mobile buffers, which provide the coupling spatially between the cytoplasmic compartment and the surface membrane. So we can summarize how we believe the pH waste disposal system operates at least in multicellular tissue. So we've built up from a subcellular to a cellular to a multicellular level. And we can see that the protons, when they're generated within a cell, are transported and shuttled to surface membrane proteins which extrude the acid from the cell. They're also shuttled spatially within the cell through gap junctions into neighboring cells, which tends to even out the pH load. And in doing so, the dissipation process reduces the mean loading in multiple cells. There are two systems. There is the dipeptide system, and there is the Ca2 bicarbonate shuttle. So this seems to be well validated. We put it through a number of tests experimentally, and the diffusive buffer system seems to describe the spatiotemporal behavior of protons within cells extremely accurately. So the next big modeling challenge, and I've got a few slides to finish on, is how does this system affect the function of the cell? I've said that the cell would close down if protons were allowed to accumulate within cardiac cells. And the effect of acid on cardiac function that I'm going to describe today will be the effect on the calcium signaling system in cardiac cells. Now, this has been modeled previously, and there are a number of conceptual models. And what I'm going to show you is not a quantitative model. What I'm going to show you is that the models that we've developed so far for the effects of acid on calcium signaling are either wrong or incomplete. There are a number of elements that we've ignored which are of immense importance. And a number of these have been staring us in the face. A number of others have only emerged when we've started to look at spatial diffusive properties of proton movement. So the basic idea is when the pH falls in a cardiac cell, acid is extruded, carbon dioxide diffuses out, they get into blood vessels and the acid is taken away. But the fall of pH in the cardiac cell, I've shown you, Gaskell showed it, depresses contraction. Contraction is activated by calcium, and we've known for 30 years that a fall of pH in cardiac cells produces a rise in the intracellular calcium levels, particularly an increase in the amplitude of the intracellular calcium transient. And part of that seems to be mediated through activation of the sodium hydrogen exchange protein, also to a lesser extent the sodium bicarbonate cotransporter. And we proposed about 20 years ago that this increase in intracellular calcium sustains contraction in the face of the acid load. So it's a cellular protective mechanism tending to maintain calcium-dependent function while protons are being removed from the site of generation. The problem is that if that calcium rise is sustained over periods of days, weeks, or months, it can lead to cardiac injury, arrhythmia, cardiac hypertrophy, and even increased progression to heart failure. So this system is of immense physiological and pathophysiological importance in the heart, which is meant that selective inhibitors of sodium hydrogen exchange like cariparide or zoniparide have been developed by the pharmaceutical industry as possible protective agents against these outcomes. What I've also shown you is that connexins spatially can confine acid loads within the heart, which would mean you would get a confinement of calcium loading, which may then produce localization of injury and arrhythmia. So the two systems are very closely interrelated. Now, how are they interrelated by sodium-hydrogen exchange? Well, if you add an extracellular weak acid, it's a permeant molecule, and in this case we're using acetate. This acidifies intracellular pH and stimulates sodium-hydrogen exchange, and the pH starts to recover. The buffering capacity is very high here, so the recovery looks quite small. And it's associated with a very large rise of intracellular sodium measured using a sodium-sensitive fluorescent dye. If you inhibit sodium-hydrogen exchange, so you're inhibiting the efflux of sodium in exchange for the efflux of protons in exchange for the influx of sodium. If you inhibit it, there's no rise of sodium. So bear that in mind. The relationship between pH and calcium is believed to occur because of functional coupling between membrane sodium-hydrogen exchange and sodium-calcium exchange, which raises calcium. So what happens? Well, we have a fall of pH in the cell. That stimulates sodium-hydrogen exchange. That raises sodium. The rise of sodium then acts on sodium-calcium exchange in the membrane, and that raises basal levels of calcium. They stimulate Ca transport pumps, which are calcium ATPase transporters on the sarcoplasmic reticulum, which sequester some of that calcium into the sarcoplasmic reticulum. They increase the release of calcium during the next action potential. That increases the amplitude of the calcium transient, and that helps to sustain inotropy in the face of the acid load. In other words, the elevated calcium starts to outcompete the protons for attachment to troponin C. So let's just look what happens if we look carefully now at that scenario. So what we've done, what we're seeing here is an experiment on a cardiac myocyte where sodium-hydrogen exchange transporters have been inhibited with cariparide. So if we add acetate to acid load the cell, you've seen it in the previous slide, you get a nice acid load that's sustained, and there's a large rise of intracellular sodium. What you find during the acid load is that the systolic level of calcium, at least in rat ventricular myocytes, is not actually affected. There is a rise in diastolic calcium, which means that the amplitude of the calcium transient, which is the difference between systolic and diastolic levels, the amplitude is reduced by about 15 to 20%. And in fact, the relaxation phase of the transient is slowed, which is typical of effects of acidosis on the time course of calcium transients. So what we see is that acidosis inhibits the calcium transient amplitude when we haven't got compensation mechanisms operating via sodium-hydrogen exchange. And this is in line with what's been published previously. But let's look what happens if you allow sodium-hydrogen exchange to operate. Well, everything looks all right. What you find under these conditions is that the systolic level of calcium climbs. We still get a rise in diastolic calcium. It's then flat. And then we get an increase, therefore, here in the amplitude of the calcium transient. So what we're doing here is we're just averaging on a slow time base individual calcium transients. And you're looking at the time course of systolic calcium and the time course of diastolic calcium. Which means if you look at the transients, they dip down initially, and then they climb up, and they climb up here, and that increase here is in parallel with the increase of intracellular sodium. So everything's fine. That looks as if it's behaving itself, until you look at what's happening to diastolic calcium. Diastolic calcium is independent of whether NHE is active or inhibited. But there's a large sodium rise occurring here, about 10 millimolar rise, and there's no sodium rise there. And this model says that raising sodium should raise calcium, which loads calcium into the SR. Well, there is no sodium-dependent rise of calcium in a cardiac myocyte when sodium-hydrogen exchange is inhibited. And we've looked at this in a whole variety of scenarios. It just doesn't happen. And this must mean that the rise of calcium that's being promoted by the rise of sodium is occurring in a compartment that is not the bulk cytoplasmic calcium compartment. And of course, it'll be the dyadic space, which is where most of the sodium-calcium exchange proteins are focused. In other words, that compartment is the one that's being controlled by the sodium that's being imported by sodium-hydrogen exchange. NHE does not control diastolic calcium, but diastolic calcium is rising. So what's controlling it? So that's what you've seen before. So we're now looking at an experiment where we inhibit sodium hydrogen exchange and we look at diastolic calcium during the acid load. And you find here, there it is, it rises. It rises actually on average by about 60 or 70 nanomolar for a pH change from 7.2 down to about 6.8. And it's reversible within a few seconds. This looks like a calcium store that's being activated. So where's the store? Well, it's not the sarcoplasmic reticulum, because if you empty the SR with caffeine and you add thapsigargin to inhibit circopumps, the proton-activated rise of diastolic calcium is even larger. But if you inhibit mitochondrial function with mitochondrial poisons, here we're using FCCP, but we've also used mixothiazole, which is a complex 3 inhibitor, you wipe out most of the diastolic rise, which suggests that mitochondria are controlling diastolic calcium. If you uncage protons within a little spot, 5 by 5 microns, deep within a cardiac cell, where you have no external sodium and no external calcium, you get a release of protons. These are line scans of protons. The length of the cardiac cell is from left to right and time is down. And this chevron shape for protons is the slow diffusion of protons out to the end of the myocyte. The time bar that you can't read there is 30 seconds. What you find is that as the protons move spatially down the length of the cardiac cell, they are followed by a trail, like a comet, of elevated calcium, and that's shown here. So now we're measuring calcium while we uncage protons in a localized spot. And the calcium ions diffuse down the length of the cell. They diffuse even slower than the protons, 10 times slower. If you inhibit circopumps with thapsigargin, the calcium that's released by protons now diffuses faster. The chevron is a little bit flatter, which indicates that the SR is modulating the proton-activated store release, but it's not the source of the release. If you add mixothiazole, 10 micromolar, most of that release, most of the calcium released by protons is eliminated. There is a small trail left. FCCP almost completely wipes it. So we are dealing with a mitochondrial store, which is a controller of diastolic calcium and which is activated by protons and releases calcium. So what we can say then is that sodium hydrogen exchange, which is very famous, modulates intracellular sodium, and that modulates the releasable calcium from the SR, probably by controlling calcium in the dyadic space. The resting bulk level of calcium is an independent calcium domain, subject to its own rules, and that's influenced during acidosis by mitochondrial release of calcium, which means if we have this model that I showed before, that not only does pH affect calcium through NHE, it affects it through mitochondria, which is why I said the original models were either wrong or incomplete. We think they're probably incomplete. Finally, just one slide to show that this mitochondrial release system, which operates locally, can produce standing gradients of calcium within cardiac myocytes. This shows a partial exposure of a myocyte to an acidic molecule, acetate, which, and this is pH, which acidifies the left end of the cell, but not the right end of the cell, because protons diffuse slowly. And we get a standing gradient, which will last for half an hour or an hour. It lasts for as long as you expose the cell to acetate. That standing gradient is not affected, for example, by a mitochondrial inhibitor like FCCP, and it's about 3/4 of a pH unit. If you measure calcium under those exact same conditions, you find that calcium is elevated by the protons in the acidic region, but not in the normal pH region. And we get a very large standing gradient of calcium. It's about 170, 180 nanomolar at one end and 100 nanomolar at the other. And that's stable for as long as you partially expose the cell to acetate. If you put on the mitochondrial inhibitor FCCP, the gradient for calcium collapses. The proton gradient remains. So this mitochondrial system is highly localized, and it can produce spatial non-uniformity of calcium in response to spatial non-uniformity of pH. Calcium maps on to the pH. What about, finally, the calcium transients, the electrically evoked release of calcium from the SR under this non-uniform pH circumstance? What we find here is if we stimulate our cells so we can get transients, we find that the transients are reduced in the acid zone, providing sodium hydrogen exchange is inhibited. Well, that's fine, because we've seen that whole cell acidosis reduces calcium signaling. The calcium transients at the far end of the cell, where the pH is normal, are normal. So what we're plotting here is the change in the amplitude of the calcium transient. So we dissociate the two ends of the cell. They behave completely independently and locally. The interesting thing about sodium hydrogen exchange is when you switch it on and you allow it to function, then the calcium transients are enhanced both in the acid zone and in the non-acidic zone. So sodium hydrogen exchange protects against non-uniformity of pH by producing a uniform enhancement of the calcium transient. But that's superimposed onto diastolic gradients of calcium, which are nothing to do with sodium hydrogen exchange and are mitochondrially engineered. So that's really all I'm going to say. The summary says that acid is extruded from cardiac cells on transporters. The transporters are coupled to the cytoplasm by means of mobile buffers, most of which are histidyl dipeptides, although interestingly neonatal cells seem to lack the dipeptides and rely almost exclusively on bicarbonate. There is a spatial control system which is proton-gated and operates through connexin gap junctional channels that tends to produce a pH syncytium in normal tissue, which evens out discrepancies spatially in the pH, but can act as an emergency safety system and shut down to permit acid extrusion during extreme acid and base loads. The influence of the system on calcium signaling and hence function is that protons tend to be inhibitory, but sodium hydrogen exchange enhances the transients, the releasable calcium, to protect normally against the inhibitory effects of acidosis. And this is assisted independently by a proton-activated calcium release from mitochondria. And the sum of the two effects produces systolic calcium. So our final diagram shows that you can get localization of pH physiologically and experimentally, and that produces remarkable localization of intracellular calcium. So I think now the challenge is to incorporate that into an integrated multicellular model. I think we've got most of the elements. Some of the mechanisms we still have yet to work out, but it's significantly different from the effects of pH on calcium that we've worked with up until now. And the people that we thank for the work, a big thanks, enormous debt owed to Pavel Sventak, who's now a joint PI in the proton transport group with me in the Burden-Sanderson Cardiac Centre in Oxford. Thanks to Peter Cole and Patricia Cameliti, who assisted with the experiments on the neonatal cardiac strands. A long-term collaborator, Ken Spitzer in the United States in Salt Lake City. Nick Smith and Steve Niederer, with whom we are now beginning to model, based on new experimental data, the kinetic parameters of some of the surface membrane transporters. And Chaehyun Lim in Seoul, Korea, with whom I did the first quantitative non-spatial model of pH regulation. Thank you very much.
Fantastic talk, Richard. I think we've only got time for maybe one or two very quick questions. So I think over there.
Very, very quick question. So Richard, really exciting new data. Very exciting. When we were looking at the effect of pH changes on calcium handling in cells using modeling, one of the things that we noticed is that an acidosis, if you look at the effect of acidosis purely on inhibiting circa, if you postulate that might happen, that we saw no change in systolic and a small rise in diastolic. Is that something that you've been able to rule out in your as a mechanism of operating?
Well, we're convinced there is a change in circa. When we have an acidosis, providing NHE is not active and producing compensation, systolic actually remains unchanged. the diastolic rises, so the releasable calcium goes down, the amplitude of the transient goes down. If we measure the calcium content after about 3 minutes of the SI using a caffeine pulse, it's the same as in control. So probably, but the release is down. So probably what's happening is the RYR channels are being inhibited partly by protons, and that's balanced by a comparable inhibition of circa. So the system remains in a steady state. If you have NHE functioning, of course, the content of the SR then goes up. There have been problems in the past. People haven't always reported this sort of effect, but many people who use weak acids and weak bases to modulate intracellular pH and measure contraction have failed to take into account that these chemicals bind large amounts of calcium. If you use 80 millimolar acetate to acidify a cell, you're binding about 20% of the calcium in the extracellular medium. And that by itself causes quite a significant decline. So you have to make sure that all your solutions are calcium balanced. When we did that, then we saw a nice flat systolic level.
Richard, I have been asked to be very strict about
Yeah.
time, so I think further questions you will have to ask Richard at coffee break. So can I now invite Christian Solar to tell us about measurements of